Okay, well, thank you all for coming. Such a beautiful day, and you could have been on the beach. You could have come in here, so I thank you all for coming. As you know, this is one of the community forums sponsored by the Board of Health. Just for your information, this is a public meeting, and the meeting will be audio and visually recorded by the town system, and as well, just for information, PBS Nova will also be filming this meeting. So tick-borne disease. We all on this island know a lot about tick-borne disease. Dr. Lepre is here. He knows more than anyone. It's a major problem, and we're always looking for ways to deal with it. Well, Dr. Elsvet has been here before, and he has this unique project of really attacking it at the point of the mouse. We all hear about the deer, but remember, the mouse, white-footed mice, play a significant role. So this meeting is open. If you have questions, I don't know how Kevin wants them during or after. It's up to him. But we just do ask you if you come down to the microphone here to ask the question, and would certainly appreciate if you would identify yourself before the question. So it's a great pleasure, Kevin. Welcome.
Thank you so much, Malcolm, and thank you all for coming here and spending your time here indoors as opposed to outside where it is particularly gorgeous today. So we're here today to tell you about this now community-directed project to try to prevent tick-borne disease. Now this is just a proposal, and it is many years from actually reducing the incidence of tick-borne disease on Nantucket, if it ever will. But it is of the utmost importance that the community take an interest in the project and, in fact, direct the project from inception. So thank you so much. And please, that means do feel free to interrupt me or ask questions during the presentation. And please come down to the mic, and hopefully I will notice and pause and not get too caught up in explaining the idea to you. So as Malcolm mentioned, the idea here is to tackle the problem of tick-borne disease at what is in many ways the source, which is not actually the ticks but the mice. That's because for almost all of the pathogens that cause the various tick-borne diseases, the ticks are not born infected. They get infected when they bite an infected mouse. So the idea is if we could immunize all of the mice indefinitely, then we could prevent most human cases of tick-borne disease. So how would you do that? Well, the idea is if we can release enough resistant mice that have heritable resistance, meaning they can pass it on to future generations of mice on the island, then that resistance would result in many fewer infected ticks and consequently fewer cases of disease. Now, this kind of resistance is actually present to some extent on the island already. That is, there has been a study that specifically looked for development of resistance in the mice on the island, on Nantucket, and found it. So what we are interested in doing is taking that adaptive resistance and making it heritable so it could be passed on to future generations of mice and ensuring that they all have it. This would require us to edit the genomes of the mice. That is, this is not something that nature normally does. We would be taking these evolved resistance elements from individual mice, and we would be encoding them into the genome so they could be passed on in a way that doesn't normally happen. Now, the reason I'm interested in this project is because I firmly believe that science takes place too much behind closed doors. And I think we need to go out and talk to communities and ensure that they tell us what they're actually interested in before we bother to even begin the research. So this project began last year when we came out here and introduced the idea, and many of you were in fact here then, and said, in effect, is this something that the community would be interested in at all? And the response was yes, at least in the laboratory, please move forwards. So part of today is to give an update on the progress that we've made, but also for those of you who were not here last time, I want to give just a general run through of the project before Joanna, who is the project manager, will come up here and give you that update. So community-driven science, frankly, what we're talking about here is altering the shared environment. And that means that we can't do this the way most science is done. Most science is done behind closed doors by scientists that generally don't go out and ask the community if they want the research done. But because the shared environment is shared, and because nobody can opt out of depending on the natural world, it would be wrong to do this kind of thing behind closed doors. So my hope is that this can be a small step towards a better model of science that is open, that is community guided from the start, that can agree on necessary safeguards and tests early on, that can be developed and run as a nonprofit, and one that can be independently monitored during the safety testing and the field trial stage, because frankly, in science, the last person you want to test a hypothesis is the person who proposed it, because they're the most biased person in the world. Similarly, the last person you want to test a technology to see whether it's safe and effective is the person who developed it. So that can't be us. We need independent researchers to run that evaluation, and we need you to help tell us what sorts of tests you would want done. To be clear, this project will only move forwards if the community supports it at every step of the way. And it does not in any way mean that you should stop doing what you're doing right now to control tick-borne disease, because nothing is going to happen for at least 6 to 7 years in the future. So by all means, try every other option on the table before then. And only then, if that's not enough, should you consider doing this sort of thing. So we now have a steering committee for the project that's been appointed by the Board of Health. And the steering committee now actually directs the research in the lab. That is, those of us in the lab, by which I mean primarily Joanna. We are the technical hands of the project, but it's the steering committee as the representatives of the Board of Health and thus the broader community of Nantucket that actually are in the driver's seat. And of course, we have a number of local and national ecologists keeping a close eye on the work, some of them on the steering committee, some of them we hope to recruit for the independent monitoring. And this is not the sort of thing where we edit the mice in the lab and then release them on Nantucket. No, 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 no. We need to figure out what might the ecological effects be. So that means an intervening field trial. Personally, I've been involved in the development of a technology called gene drive, which allows a alteration to spread through a wild population. That is not what we are proposing here. That would, in fact, be a terrible idea in this case, in my opinion. It's the sort of thing where we're working on versions that do not spread indefinitely and could be local and might eventually help the mainland if it goes through here. But that is not what we're doing here. So this would only release mice that were edited, and that's the end of it. If one of them catches the ferry to the mainland, then that immunity would not spread on the mainland. Some rules that I think about when engineering complex systems. Because if you work with biology, you learn we don't understand everything. There is no way that we will ever understand everything and be able to perfectly predict what will happen because things are just too complicated. So whatever the problem is, if you have a problem that you need to solve, you should always start by trying to make the smallest possible change that you think can solve the problem. Because again, you don't know what the effects will be. And again, since you don't know for sure what the effects will be, you want to start small, try to find out what they are, and only if it looks good would you then scale up to the next level. So that's why we'd start in the lab, then a small field trial, then eventually maybe moving up to Nantucket. So the reason why we might want to alter the environment for this project is because tick-borne disease is an ecological problem. So for all of the major pathogens, they're passed back and forth between mice and ticks. A tick that bites an infected mouse gets infected. It then bites an uninfected mouse and infects that mouse. There are other reservoirs, but the mice are the main one. This would not concern us particularly because, among other things, the mice actually don't get disease, as best we can tell. But if we get bitten by an infected tick, then we do. So this is the problem. The idea of this project then is to say, well, what if the mice could no longer be infected? That would remove the primary reservoir of the disease. You would have many fewer infected ticks and therefore many fewer infected people. We can't promise that if this works perfectly in the sense that no mouse can ever become infected, that would not reduce the disease rates to zero. That's just not possible. These pathogens are in the environment. They're in other species apart from the mice. You're never going to get rid of them entirely, but there would be a lot fewer cases than today, we believe. How do you do this? Well, again, Joanna will tell a little bit more about this, but when we get sick, our bodies learn to recognize the particular molecular signature of the pathogen. And over time, our immune system evolves a response that can pinpoint it precisely in order to attack and destroy it. These are antibodies, or the molecules that can do this. So the idea is, if every mouse, instead of having to be exposed first in order to make antibodies, if they all produced protective antibodies that targeted those pathogens, or as Joanna will talk about later, the tick itself, and so that they were immune from birth rather than having to be exposed, then we think this would break the cycle of transmission and prevent tick-borne disease. Again, it would not go down to zero, but we believe that it could be a very considerable reduction. How much? Well, that's the sort of thing the field trial would need to determine. And we have a wonderful team that we've put together, and I'm going to hand it over to Joanna to tell us a little bit more about the technical details of how this project works, and also to give an update on results, because she's been staying up late nights for the last several months to tell you this.
Including last night. Could I ask a question before you switch?
Yeah.
If you assume, for instance, 10,000 people on Nantucket annually, how many of us currently are getting tick-borne diseases?
Annually, I'm going to have to defer to the members of the Board of Health for that one. Malcolm?
Well, maybe Dr. Lepre, I mean, a lot of surveys have been done, and I would say about 40% of the island at various times has had a tick-borne disease.
Okay.
That's about the numbers I can give you. And year to year, it varies.
Thank you.
Mm-hmm. Other questions? All right. Joanna.
Two years ago, Dr. Esvelt had an audacious idea: protect people from the scourge of tick-borne disease by endowing the little mice that harbor these diseases with the power to defend themselves. Today, his idea is being realized by a diverse team of researchers at MIT, Tufts, Harvard, ArcBio, and the Broad, working together to create white-footed mice that are immune to tick-borne disease and capable of passing that resistance to their offspring. Our team includes infectious disease experts, molecular biologists, antibody engineers, computer scientists, and a bioethicist. Our research requires a highly collaborative effort across a wide range of disciplines and cutting-edge technology in order to engineer an organism that is, at least genetically, a black box. So we hired Dovetail Genomics to sequence and assemble the very first white-footed mouse genome. However, before we could start working with this genome, we needed to be able to make sense of it. And unfortunately, we couldn't easily look to another species for a biological roadmap. The well-studied house mouse is a very distant relative. The house mouse and the white-footed mouse haven't shared a common ancestor in 25 million years. In fact, the house mouse is more closely related to the rat than the white-footed mouse. Because of its unique biology, the process of engineering this mouse has been as humbling as it's been rewarding. The following technical summary only skims over our many misdirections, all the twists and turns in our scientific journey, but let me tell you, we tried countless techniques. We sought guidance from an even larger group of experts, and we spent many sleepless nights troubleshooting in order to finally discover some extremely tiny and yet incredibly potent antibody genes. So how did this all come about? Luckily, Dr. Esvelt didn't have to invent mouse superpowers to prevent tick-borne disease. He could immunize mice with proven vaccines and harness the mouse immune system to create specialized antibodies against the pathogens that cause tick-borne disease. The problem is there are tens of thousands of mice on Nantucket, and Dr. Esvelt needed a new strategy to immunize mice en masse. He combined the power of the immune system with the tools of synthetic biology to devise a genetic therapy that could provide heritable, long-lasting immunity. See, antibodies are normally created from scratch by each new organism's immune system. Dr. Esvelt envisioned creating mice with anti-tick and anti-Lyme antibodies that would be present from birth and provide lifelong protection. This is actually pretty innovative. We will be the first scientists to turn the immune system's individualized protection into a trait that can be inherited. So we're researching two different types of antibodies. Our initial work has focused on an antibody that protects mice from the pathogen, the bacteria that causes Lyme disease. But we will soon begin research on another type of antibody that inhibits tick feeding and therefore prevents, blocks transfer of many more pathogens to and from white-footed mice. This second type of antibody should have an impact on the spread of anaplasma, babesia, ehrlichia, and many other pathogens, all the other pathogens that are carried by blackleg ticks. As an added bonus, it should substantially reduce the tick population and return tick levels to those of a century ago. So, very, very generally speaking, engineering a tick-borne disease-resistant white-footed mouse involves vaccinating mice, isolating the most protective mouse antibodies, and integrating those antibodies into the mouse genome. In the first step, Dr. Telford vaccinates mice with the Lyme vaccine to have those mice produce anti-Lyme antibodies. Dr. Telford repeatedly injects a very small group of mice from his colony with a portion of the bacteria that causes Lyme disease. The mouse immune system identifies a foreign body, and mounts an immune response. With each additional dose, the mouse immune system creates progressively better anti-Lyme antibodies. After many injections, Dr. Telford checks the mouse blood for the presence of great anti-Lyme antibodies using a test called an ELISA. In the second step, Byong isolates B cells that produce anti-Lyme antibodies. In order to integrate these antibodies into the mouse genome, we need the DNA sequences that code for them. These DNA sequences can be found within B cells. In order to isolate these B cells, Byong first extracts white blood cells from the spleens of Dr. Telford's vaccinated mice, and then he places these cells in a machine called a flow cytometer that differentiates B cells with anti-Lyme antibodies on their cell surface. I believe that we are the first team to isolate B cells from white-footed mice. This process was complicated by the fact that nobody had any idea what their B cells look like. We couldn't just rely on normal protocols because our mice are so different from other species. A combination of luck, an old research paper, and a very important piece of advice from Dr. Telford led to this accomplishment. In the third step, we sequence the DNA from the B cells that make these anti-Lyme antibodies. Once we've isolated these B cells, we submit samples to MIT's Biomicrocenter for deep sequencing. In the fourth step, CC uses bioinformatics to identify the anti-Lyme antibody sequences. But before CC can start searching for these sequences, she needs to piece together the many truncated sequences that are produced from the sequencing machine. She does this by drawing on the Broad's computational power to assemble tens of millions of sequences from multiple B cells with many computers working in parallel. And then next, she uses search algorithms to hunt down our antibody genes. This is harder than it sounds. In the final step, we integrate these antibody genes into the mouse genome using CRISPR. This phase of the project combines bioinformatics and synthetic biology. First, we're going to work with CC and other computational biologists to identify safe locations in the mouse genome. Next, we'll package our antibody gene sequences with the CRISPR machinery and deliver all of these components into mouse reproductive cells. The CRISPR machinery will cut the mouse genome at precise locations and initiate a cellular repair process that will integrate our antibody genes. I need to reiterate, we are not building a gene drive for Nantucket. We're using CRISPR as a molecular scalpel to insert our antibodies, but CRISPR itself will not be integrated into the mouse genome. Without CRISPR, our mice will lack the essential component of a gene drive. In order to ensure that our antibodies are passed down for many generations, we plan to encode many copies of these gene sequences into the mouse genome. So according to the fundamental laws of inheritance, a parent mouse has a 50% chance of passing any one of its genes to its offspring. However, by encoding tens to maybe even hundreds of copies of our antibody genes, by scattering these throughout the genome, we can virtually guarantee that all offspring and many subsequent generations inherit our antibodies without necessitating a gene drive. When we last visited Nantucket in January, our work had only just begun. We reported that Dr. Telford had been vaccinating mice from his colony with the Lyme vaccine. Since then, we have isolated B cells with anti-Lyme antibodies. We sequenced those B cells, and we identified unique antibody genes, these anti-Lyme antibody genes. Over the next 6 months, we will reproduce these antibodies from their gene sequences, and we will test their efficacy before putting them in mice. As promised, we will also begin the process of isolating that second type of antibody that inhibits tick feeding and therefore protects mice from many more pathogens. My husband likes to say that this second type of antibody makes mouse blood taste like super hot Tabasco sauce for ticks. So we're going to figure out the strength of our antibody hot sauce, and we'll let you know. I am very personally motivated to see the end of tick-borne disease. My family and I are residents of Chilmark, the epicenter of tick-borne disease on Martha's Vineyard. I have 3 young kids, ages 3, 2, and almost 1. This past weekend, I watched as all 3 of my kids played outside together for the first time in the grass. The older 2 ran through the sprinkler while the little one just crawled around. I wish I could say that I shared their joy, but I spent the entire time worried about ticks. I can promise you, we will do our best to immunize your mice, and hopefully one day we will all be able to enjoy the outdoors without worrying about ticks.
So thank you, Joanna. She makes far too much of my contribution. This has really been her project, and she's really been driving it forwards. And also, Sam, it would not be possible without you. And to return to the future, so assuming that we can move forwards and make mice whose blood tastes like super hot Tabasco sauce to ticks, then what would that mean? So we're giving ourselves maybe up to 2 years. Hopefully it shouldn't take nearly that long. We're giving ourselves up to 2 years to successfully do that. As Joanna mentioned, white-footed mouse, not exactly a model organism. It's still a mouse. CRISPR works in pretty much everything, but no one's ever edited the white-footed mouse genome before. So now we know what the sequence is, but we still need to figure out how to do that. Most of the standard methods require you to be able to work with mouse embryos and implant them into pseudo-pregnant animals and so on. None of this has been worked out for the white-footed mouse. So we're researching some alternatives. We'll hopefully won't have to do that from scratch, but that is an unknown. The reason why we were willing to go to you initially last year with this project, though, is because I always say that saying in science, standing on the shoulders of giants. This is a engineering project in the sense that we're confident that it can be done. We're not sure how long it will take because things fail in science. In fact, most experiments fail. But we're confident that we can do it. And so we didn't want to come and waste your time until we were confident that we could actually do it. Again, timeline is difficult. But our best guess is 2 years from now, hopefully sooner, now that we've made progress, to really confer heritable, passed on to descendants, immunity on mice in the lab to both, ideally, the Lyme pathogen Borrelia and also against the tick saliva. Once we have those, then it's a time for a field trial. So we've been talking to various groups, both private and public, that have small islands, preferably mostly uninhabited, around this area, in fact, up and down the coast in numerous states, that could potentially be candidates. And most of them would prefer that we not, or at least the private ones would prefer that we not say which ones they are until it's shown that they're actually good candidates. So what does that mean? That means that we're going to rely on Sam and others to go there, and because Sam is really the world's leading expert in tick-borne disease in these islands for sure, and tell us, are those islands a good match for Nantucket? Because we need to know that the ecosystem there is similar enough to the one here that if we were to release these mice on the island, then that would be a good proxy for what would happen if we were to release the mice on Nantucket. So we need to ensure that they're a good match. That also means we want to hear from all of you, and really from anyone anywhere in the world that has particular concerns about things that would need to be looked at. Because not only do we need to know what should we look at during the field trial, once mice are released onto the small island, but we also need to know, are there any particular criteria that you're worried about that you think might be unique to Nantucket or might not be present on the island, so that we could then use for the evaluation when choosing which island is most suitable. The challenging part is, even once we have an island selected, we need, of course, regulatory approval. So we haven't, right now, we view this project as community-driven. Your criteria for release and moving forwards at every stage is probably going to be way more stringent than what the various regulatory agencies are going to require, or at least that's our operating assumption. And morally speaking, you're the ones that live here. It's really up to you. So we're more concerned by what you think than what the regulators think. However, there's this little problem in that if we do something without their permission, then we get arrested and thrown in jail. So we have to go. So we're going to go and talk to them. And this is a little bit of messy because if you believe it or not, if you ask who would regulate this, the answer is nobody knows. It is probably the Food and Drug Administration, because technically if you take a mouse, even if you take pre-existing white-footed mouse genes, encode them back in the white-footed mouse genome in different places, to the FDA, that is a veterinary drug because you are conferring a trait that did not exist on the mouse before. That is, the DNA, even though it originally came from a mouse, still is a veterinary drug because you change the traits of the mice and their descendants. Even though drugs don't normally get passed on to descendants, that's the way the law is written. So it's probably the FDA. However, you can imagine how much the Food and Drug Administration knows about ecosystems. So, more than you'd think, because they actually had to do the Aqua Advantage salmon recently and the controversial Oxitec mosquitoes in Key West, Florida, and a number of others. But even so, they're partly due to those experiences, they've decided that maybe perhaps we should defer this to either EPA or Fish and Wildlife. So we're going to talk to all of them and see what they would want to require as well. Because in particular, Fish and Wildlife has a lot of expert ecologists that could hopefully help with this question of which island is most suitable and what do we need to look for as well. But we have to breed up enough mice from the initial immune ones in the lab to release on to the small island, which is probably several thousand mice. And I'm hoping that Joanna is right and we can engineer them so as to scatter dozens, or maybe even if things go wildly, miraculously well beyond my wildest dreams, hundreds of copies in the genome. Then it will be pretty easy. But in all likelihood, we will definitely have to ensure that the mice are a perfect genetic match for the ones already there, which means taking the ones in the lab and then capturing wild ones from the target island and then crossing them. And every time you do that cross, you lose half of the resistance genes. So in order to ensure that it's a perfect background, we're going to lose a lot of those resistance genes, which means we're going to have to constantly adding new copies to make up for that. But we eventually need a population that has, well, lots of resistance genes, but is also a perfect match and is reasonably fit, because we're starting from Sam's mice, which have been in captivity for 21 generations. So that probably means they've accumulated some variants that mean they're well adapted to Sam's laboratory and not so much to the wild anymore. But by crossing them back, we should get rid of those and they should be more fit for the wild. So once we've done that, that's going to take a few years as well. Regulatory approval permitting, community go-ahead permitting at that point. We would then begin releasing on that island. And then in order to determine how effective this is, we need to know primarily, do mouse infection rates go down? But the real question is, do tick infection rates go down? And the tick generation time is 2 years. So you want to know how effective it is, you really need to wait at least 2 years. Arguably a little bit less than that maybe, but you really want to probably wait at least 2 years to see what's going on. And of course, ecosystem, right? We don't understand the details, so we need to make sure that nothing else goes wrong, nothing else that might be on some kind of time delay that we don't understand. So that's going to take another few years, which means you're looking at probably on the order of 7 years, or at least 6.5 at the absolute earliest, probably more like 7.5 before you could even be in a position to consider a release on Nantucket. So that's what I mean when I say, whatever you're doing now to stop tick-borne disease, don't stop, because this is quite a few years in the future. How would this work? Again, if you decide to move ahead with releasing mice on Nantucket, well, as Joanna mentioned, there's maybe 50,000-ish mice, and again, Sam is the expert here, so correct me if I'm wrong. There's maybe 50,000-ish mice on the island at the end of winter, because that's the low point. And at the high point, at the beginning of winter, towards the end of fall, then there might be as many as 500,000. So that's the range we're talking about. Since we want to release mice that have as many copies of the resistance gene as we can get, then we need to release enough so that they reproduce effectively enough that, on average, every mouse has at least one copy. Which means we want to release at the low point, so at the end of winter. And we still need enough. How many will depend on how many copies of the resistance genes we can put in those mice, the mouse genome. So that could vary by a factor of 100 if we're really lucky. But it might mean releasing, say, 100,000 mice on Nantucket, which is a lot of mice. Like, let's be real, 100,000 mice is a lot of mice. Can we even grow 100,000 mice? Can we breed up that many? Well, Jackson Labs, which is this nonprofit in Maine that supplies a lot of the research labs with house mice, they produce on the order of 3 million mice every year. So how to do this is known. White-footed mice are a little bit feistier, but still can definitely be done. And you release that many, then you're going from 50,000 to 150,000, except, of course, that every predator on the island says, ooh, all these mice that don't know when to duck and cover. Yum! So you're going to see some losses to that, but frankly, that's enough mice at that time of year when there aren't normally that many mice, so the predators are mostly going to eat their fill, at least for a while, and you're still going to have a lot of mice left over, and they're going to hopefully reproduce and breed with the wild mice, and there will hopefully be enough of them left, and again, this is the sort of thing that would require monitoring, that most of the mice would have these resistance genes, and it would just require that one big release. And of course, it gets better the lower we can reduce the population from that. If that's normally a low of 50,000, if we can set traps to reduce that initially, anything the community can do, then the more potent the effect. And the fewer mice we might have to release, the less likely we might need a second release. So this project now has been going on for a little over a year now, and we're working with the Vineyard as well. They have their own separate steering committee, and we've presented at a number of times both here and on the Vineyard, and now both islands have steering committees with full membership, and the Nantucket steering committee has currently 6 individuals on it, and I think we're all here. So Danica Connors is a Nantucket herbalist, and native who helps people recover from Lyme disease. Howard Dickler is an MD. There's Howard, who ran a division at the National Institute of Allergy and Infectious Disease. And, well, for some reason, the Board of Health, in their infinite wisdom, decided to put me on the steering committee, which I'm not sure is appropriate, but Malcolm, yeah. You can always fire me from the steering committee. It's okay. I won't be offended, I promise. We have Dr. John Goldman, here's John, who is the editor of the Journal of Infectious Disease and a retired professor of immunology. And we have Dr. Emily Goldstein-Murphy, who is right there, hasn't had to leave yet. So Emily is at Mariah Mitchell, and she's the local rodent expert on the island. And, of course, we have Dr. Sam Telford, who's a professor at Tufts Veterinary, who's, again, been studying tick-borne disease on Nantucket for 30 years. And, of course, we have Joanna, who is really driving the project forwards and is the project manager. But, of course, we all, okay, not everyone on the steering committee, but we at least have a stake in this project, which means you should not trust our judgment as to whether or not this is a good idea. That means we need, in addition to the steering committee, because I imagine that by the time we get to the point of evaluating the field trial results, most of the steering committee might be somewhat invested as well, which means to some extent you shouldn't trust them either. You need a group of independent experts that have nothing to do with the project, but do have the expertise to evaluate the results. So in drug testing in pharma, they have what's called the Data Safety Monitoring Board to monitor the clinical trial in case anything goes wrong and help evaluate the data and so on. What we're envisioning is something even more substantial than that, putting together a group of independent scientists and experts from other disciplines to oversee the trial and evaluate the data and deliver a recommendation directly to the Board of Health and the community at the end. We don't know how to put this body together because no one's ever done anything like this before. So suggestions would be very much appreciated, but one option would, for example, involve asking the National Academies of Science and Engineering and Medicine to put together a panel of experts, because this is the sort of thing that they do. There are other options, and agencies that could do this. The American Academy of Arts and Sciences might be a good choice, but we're still exploring that, and again, suggestions very much appreciated, because, lest I remind you again, you are in charge. What does it mean in terms of decision points? Again, given that you are in charge, if at any point this looks like a bad idea, the Board of Health and the Steering Committee can just say no, and that's that. If you think this is a bad idea, then the main decision points are after we've made the mice in the lab, then there needs to be a go or no-go recommendation, ideally by the independent monitoring board at that point. And if go, then we need the regulatory approval. We need approval to release on the mostly uninhabited island, and we need continued interest by the Board of Health. The second evaluation point would be after that field trial. Then, again, the independent panel of experts would need to evaluate the results and deliver their recommendation to the community as to whether to move forwards. And my understanding, and again, this depends entirely on your civic governance, but my understanding is that there would be a warrant proposed by the Board of Health at the town meeting, and then that warrant would have to be approved by a vote at the town meeting in order for the project to move forwards. And again, this would be at the earliest six years from now, probably more. So who's supporting us? I mean, a number of other things you have to focus on, even if a project is nonprofit. Who has a stake in it? So initially, because my lab is now only a year and a half old at MIT, we're running on MIT support and some from Tufts, courtesy of Sam. We have a grant from the Greenwell Foundation, which is interested in this question of how do you ensure that a project like this is ethical? So we're working with a philosopher and ethicist, Jantine Lunshoff. So she's supported by the Greenwell Foundation, trying to, and so she wasn't able to be here today, but she's been here. She was here for the last meeting and will be here in future. And she may want to talk with some of you to see what you think Because the idea is, since no one's done anything like this before, but this is what we want to see science as like, this is how we want to see science directed, is by communities, telling scientists what they want and what they're concerned about, and the scientists changing the plan, changing the technology at the earliest stages in response to those concerns. So that's what we want. How do you develop a framework for that and encourage other scientists to do that? That's the point of that particular project grant. We have some support from the Rainwater Foundation, which has some interest in Nantucket. We have been recommended for a federal grant on Tickborne Disease Research Idea Award. And we will need, eventually, philanthropic funding for the scale-up. Because the really expensive part is not the laboratory research, although laboratory research is not inexpensive. It is really the breeding up thousands, and especially tens of thousands of mice, releasing them, and then monitoring afterwards, and looking for everything that all of you think might go wrong. That probably means some figure in the millions. And again, that would need to be raised philanthropically, probably from people who are interested in health on the islands. We don't think that that would conflict with other potential projects of merit on the island, given the unique nature of this project and ideally trend-setting in terms of how science is done in the future. But that is a potentially valid concern for any of you who think about that. So just to finish, questions for you after this. Questions for us, but questions for you. Is this one you still wish to pursue? This is a question you get very tired of, because every time I come here, I'm going to ask it. That's what we need to know. It would be painful for us, especially for Joanna, at this point, if you said no. But the reality is, if you say no, we walk away. That's the way it has to be. You're in charge here, not us. Do you want the mice to be immunized against Lyme alone, against ticks, or both? So far, the general, pretty overwhelming consensus at every previous meeting has been, if you're going to do it at all, you may as well do both. So that's sort of the current operating plan. But if you change your mind, let us know. What do you want us to look for? What do you want the independent panel to look for in terms of things that might go wrong? What are the criteria that would be necessary to release? What would make you comfortable enough to try this, if anything? Maybe there's nothing. That's fine too. Do you have any suggestions on the project governance? Other than maybe me being on the steering committee. But you can outvote me. And what should this be called? I mean, I kind of like the name Mice Against Ticks, because this is really about enlisting the mice to fight tick-borne disease. But again, it's your project. So if you want it to have a particular name, name it. This is the most important part. I believe we have a question box in the back, because I recognize that there are certain cameras around the room, and people might not want to stand up in front of the cameras and ask your question. So please, either anonymously or not, we will post answers to any anonymous questions on our site at responsivescience.org. And feel free to contact me or Joanna, or Emily has generously volunteered to have us put her work email up here so you can bombard her with questions. And I believe Danica has also volunteered. She has expressed her skepticism publicly of this project. And I think that's wonderful, because she has made it very clear that the only way that she will support this project going forwards is if everything is done correctly, which is why I'm very glad that she's on the steering committee, because she will therefore work to ensure that that is so. So please feel free to contact her as well. And that is it. In terms of questions, lest the barrage overwhelm me, I'm going to invite Tim and Sam to please come and help answer questions, because my expertise is in molecular biology. And really, I didn't have nearly as big of a role in this project as Joanna implied. So, and Tim, if you could help answer anything medical, and Sam, you know way more about ticks and disease and ecology than I ever will. And then please, questions. Please come to the microphone. Sorry. sit stand? These are acronyms. There we go.
Okay. Kevin, I enjoyed that. Joanna enjoyed the presentations. A couple of technical questions. If maybe they're too technical, we can do it later. I'm pretty good at boring people with technical questions. I presume for this to work, especially in the long run, that these will have to self-perpetuate through the population. It wasn't clear to me from the presentations how that would happen. That's my first.
Yeah. So great point. Whenever we edit an organism, whether it's through traditional selective breeding or by modern genome editing, in general, we're altering it for our benefit, not its reproductive benefit. To the best of my knowledge, there's never been a case where a human-altered organism was more able to reproduce in its natural habitat than the natural versions. So we don't expect this immunity to help the mice in anything other than a general well-being sense. That is, if I were a mouse, I would rather not have to live with ticks biting me all the time than the current state. But we know that it does not affect their reproduction, right?
That Lyme disease doesn't. The ticks themselves, if there are too many on a mouse, they certainly can get anemia. So there may be a fitness benefit there.
So there may be some selective advantage
There
with the
may
treated
be,
mice.
There may be.
There may be. The only study I've seen actually crazily suggested that slightly higher male mating success upon high tick infestation levels and no effect on females, which I read that and was like, no way. But it's possible. That said, it would be the first known example of a fitness benefit. So we don't expect this to spread on its own through the population. So the idea is just to release enough mice carrying enough copies that on average, every mouse inherits one. And even though the mice can have maybe 5 generations per year, the selective disadvantage is going to be comparatively tiny because we don't need to produce very much antibody to produce resistance. We're not going to be taxing the mouse's system. In general, organisms like us we're extremely, shall we say, wasteful with energy relative to a microbe, which has to be very parsimonious.
Yeah.
So we don't, it's just going to be sheer inertia in releasing that many mice, which is necessary if we're not going to use a gene drive.
You have to do it to find out. If there's even a small selective disadvantage, it would quickly die out.
Quickly is not the word that I would use. It might die out in a few decades, at which point we could release more.
Quantitatively there. Is the antibody that the treated mice would make precisely the same as the natural immunization? And is it polyclonal, monoclonal, so forth?
So the idea, so polyclonal is when you vaccinate an individual, then it generates lots of different antibodies. And in this case, monoclonal is when you isolate just one. So here we've isolated monoclonal antibodies from different exposed mice. And the idea is we would integrate the genes encoding those antibodies, several of them, into the genome. So you'd have multiple monoclonal antibodies. Guesstimate is probably 3 to 4 against the Lyme pathogen and probably 5 to 6 against the tick saliva protein. And that would be, and it would be the exact sequences. Because again, part of the general community opinion seems to be that if at all possible, don't add any DNA that was not from a white-footed mouse. So we will be sticking to that.
Do they make a full intact antibody with heavy and light chains, constant and variable regions, and so forth?
Yep.
It's all there.
It's all there.
With all of them.
Yep. They don't seem. They're very different from mice, but they're still mammals. It looks like a standard mammal. It's not like crazy or even like some of the strange mammals like camels.
The constitution is very conserved, and so is the light chain. The heavy chains change quite dramatically.
Okay. I'll bore you selectively later. Not everybody.
Great. Thank you, John.
Hi. So I have 2 questions. My first question is, could you apply the same antibody approach of putting a small dose of Lyme into a human and taking those antibody genes and using CRISPR to put into a human?
That's a great question. In theory, you could. In practice, so there is a technology that's being developed that actually somewhat inspired this project. It's called vectored immunoprophylaxis, which is a mouthful. But the idea is exactly that. You use a gene therapy vector to deliver the genes encoding known protective antibodies into the muscle cells, which then produce it and render you immune. This is actually what I think is our most promising defense against potential pandemic viruses. Because with this, you could isolate the antibodies from the first people that come down with the new virus, especially the ones that survive, figure out the most effective ones, and then just give everybody that particular version of immunity. And so that's the sort of thing that we could scale up conceivably inside of a month to 1 billion doses, which would end the threat of pandemic viruses. So it's that exactly what you described. In terms of other potential solutions, there is a mass biologics project that has isolated human antibodies against the Lyme pathogen, and they hope to stabilize them somehow in a way that I don't understand, that would allow them to last for months in your circulation. So you would get an intravenous infusion of antibodies in the spring that would protect you through tick season, and then you would get that every year. So that sort of thing is very much viable.
Very cool. Thanks. And maybe I missed it. I was here for the first 10 minutes. What would it mean for humans if you made mice immune to, say, just ticks rather than Lyme, or vice versa?
So if the anti-tick approach is effective, it should block Lyme probably as effectively as the anti-Lyme antibodies. That's really the safe way to go. But we know that immunizing mice against ticks is more difficult than immunizing them against Lyme disease alone. That is, the vaccine produces resistance in 50%-ish of the mice to ticks. The anti-tick one does, right?
We've been working on a first generation of this with baited vaccines. And this is the same mechanism for the old human Lyme disease vaccine. It's the same immunity that was stimulated by the human Lyme disease vaccine and the one that you give your dogs. It's generated against a protein that is found on the bacteria within the tick. And it's only in the tick. It modulates the relationship between the bacteria and the gut of the tick. And it's not found in the animal, in the vertebrate host. And so the immunity acts by coming into the tick with the blood as the tick is feeding, and it kills the bacteria within the gut of the tick. And so the human Lyme disease vaccine was 60 to 80% effective. The dog vaccine is similarly effective, and in our hands in the laboratory with mice, we can get 100% protection using anti-OSPE immunity. So I certainly think that that level of protection is easily feasible with Kevin's approach.
And we also have the advantage of even if a vaccine is not very effective, say even if it's 50% effective, then in the 50% of mice that are protected, we can test them, see how resistant they are, and then sequence the antibodies from the most resistant mice. And then we'll take the winner from each of the most resistant mice, and we'll encode all of those into the genome so they'll be able to produce the best of all of them. So we think that will make it more effective than any individual vaccine. It's the difference between saying, okay, everyone has to come up with their own solution, and testing a bunch of people and taking the best several solutions and packing them all in so everybody's protected with all of them. That also makes it more evolutionarily stable. That is, we don't know, theoretically evolution always wins in the end. But if we attack it from enough different angles, it will be very hard for it to escape all of them at once. So by adding in antibodies that bind to many different parts of the target molecule, if it wants to escape all of them, it needs to mutate all of them. So that's our best protection against either the Lyme pathogen or the tick evolving. I am less concerned about the tick evolving just because the tick generation time is 2 years. So it's going to take them a very long time to evolve away from anything we do. Yeah. Great question.
To completely eradicate the disease on this island, are we going to have to do the same thing with deer? It's called deer tick, so it seems to me we might have to take the next step.
Well, for one thing, there's no complete eradication. Lyme disease will never disappear. It's been here long before the ice ages. It was probably in the world 25 million years ago. It's been here a long time. What we want to do is reduce the risk to the point where some of your parents or their parents remember a time when they didn't have to worry about deer ticks. They didn't see Lyme disease. It was hidden away in little pockets. And so the idea is to suppress transmission to the point where we go back to that wonderful time and we're very much less likely to see an infected tick. But the word eradicate, wiping it off the face of this earth, is just not possible. But you're absolutely right. Kevin has repeatedly stressed that we should continue to do what we're doing and do more in what we call integrated approach. We need deer reduction just as much as we need insecticides as personal protection and hopefully mice that no longer cooperate with the life cycle of the bacteria.
So the operating hypothesis is this will reduce the amount of tick infected with the pathogen substantially, 95%, or?
Well, right now, I've been out in the field for the past couple days. You would encounter an infected tick every 10 minutes, let's say. So what you want to do is try to bring it down to an infected tick every 100 minutes. A 10-fold reduction or a 100-fold reduction would be even better.
Great.
And yeah, the goal is 100-fold reduction, but we just don't know yet.
Right. Okay.
Deer reduction has been tried in 2005. I sponsored it. It did not meet with universal approval, I should say. So I think the methods that have been mentioned, I mean, tick checks, find a good friend, significant other, check yourself for ticks, look at the brush, cut that all down. I think reducing the deer herd, but I think I think to eradicate the deer herd is unrealistic.
Yep. Okay. Thank you very much for your great work.
I have a question. It's probably kind of a silly little question, but you didn't mention anything about a stopgap on the population explosion of the mouse itself. I mean, the mortality rate down here, predation would be next to nothing, and if the reproduction cycle is like 44 days, and gestation is like 22 to 28, and 9 in the litter. It seems like the population would just be astronomical after a certain period of time. Is there any, have you addressed that or looked at that at all? Or?
So that would actually be a wonderful thing to discover, but we hope we would discover that in the field trial stage.
Well, the reason I brought it up is when you were giving your presentation, you mentioned the first year about a 300% increase in population, but is that real? I mean,
So assume there's 50,000 mice there, we would then, again, depending on how many copies of the resistance gene we can insert, because a mouse that has 5 copies, you release that mouse and it mates. That's as effective as releasing 5 mice that each have 1 copy, all of which mate. So the more copies we can add to the mice that we're going to be releasing, then the fewer mice we need to release.
But what happens to the actual population, the mouse population itself? I mean, that's just going to keep growing and growing and growing.
Actually, no. The first 20 years of my work out here, I would come down every year for 20 years, 6 months out of the year, 3 days a month, and trap mice to get precise population estimates. So the figures that Kevin's working with are extrapolated from what I have found to be typical mouse densities extremes. I have never seen a population go more than about 70 per hectare, so roughly 35 per acre. And this is over 20 years of intensive mouse demographic data that we have. So something is keeping the mice in check. And so if the wild mice don't explode more than that level, and these were 2 unusual years in 20 years. Typically, it's about 35 to 40 animals per hectare. Something is out there that's regulating them. Disease is regulating them. There's a mange mite that is regulating them. It could be a weather. We know that reproduction usually takes place in June, but in warm weather, they'll breed through the winter. If you have a severe winter, that will decimate the mouse population in the spring, and then you have what we call a founder effect. You have very few mice giving rise to the rest of the population, and it takes a couple of years for them to recover from that. So the mice that may be introduced as that have been the focus of this work will never be as good as the mice that you have now, and we don't see that problem.
Okay, thank you.
I should also point out that if you're concerned about mouse levels, then setting out traps near where people live and don't want the mice is not going to impact the project at all.
Didn't you state that you might possibly introduce 500,000 mice into the system down there?
No, up to 100,000 at most, which would be twice as many as would be there, shall we say, normally at that time of year. And so the normal mouse population, I'm going to say typical year is 50,000 at the end of winter when it's the lowest, and then it might rise as high as 500,000 at the end of fall. And so we would release.
Yeah, but that 500,000 would be the result of whatever was around in the spring. And so the strategy is to minimize the number of mice that need to be released by being clever about looking at the data from the past, saying, okay, the mouse population is at a very low level, typically in April, where sometimes I'm lucky to get one per acre. And so you target those periods, and you can augment that natural effect of a severe winter by going out there and trapping the devil out of the place with snap traps or with bait stations. And then once you've determined that you've lowered the mouse population even more, then you introduce some new mice. And those are the ones that will preferentially reproduce. They'll be more common. And so I think that we can get away with introducing far fewer.
Yeah. So again, I want to give you the maximum number, and we're hoping we can knock that down through a lot of different means. So, for example, if you could knock it down, say, threefold, then the ones that we would release would then bring it up to what it would normally be like.
Thank you.
And also, the female deer tick is probably laying close to 2,000 eggs. So I've gotten 170 of them off a deer. So you figure those numbers. I'm surprised we're not ankle deep in them.
Yeah, something is regulating populations out there, and so I continue to argue with Tim and I continue to argue that we need these complementary methods. We go after the source of infection, which is the mice, and the source of the ticks, which is deer. You reduce the number of ticks, and you reduce the capacity of the mice to infect those ticks, and both of them working at once will bring the number of infected ticks down to significantly lower than what it is now.
Oh, and Sam, you said today that you think that probably about 80% of ticks feed on white-footed mice.
The larval and nymphal stages, it's very likely here that's true.
Maybe not on the mainland, but here, which would mean that if the mouse blood suddenly tastes like super hot Tabasco, then the ticks fall off the mice. Most ticks don't find a host. So then
That's
most
right.
of the ones that would then try to bite a mouse will never find a host and will die, meaning there should also be about 80% fewer ticks that would be able to bite you. Even fewer when it comes to adults, because we'd be adding bottlenecks at the earlier stages of the life cycle. So one benefit of that anti-subalissin antibody, anti-tick antibody would be fewer ticks in the environment to bite you in the first place. I mean, if they don't spread disease, it's not that bad, but frankly, no thanks.
My question is about the go, no-go decision points you laid out. What information would the community have at the first point other than that you're capable of producing the needed number? I'm wondering why it wouldn't be at the second point when you're about to start field trials.
So the reason why we would be interested in... So we are unlikely to gain any more information from when we start breeding up mice for the field trial and
Right.
when we begin the field trial. So
what's the go, no-go decision then?
So that's really here is our data on how effective these mice are at resisting tick bites and at resisting infection with the Lyme pathogen. Is that good enough? And so again, we would have the independent group would evaluate the data and say, does this look real? Are we confident in this data? And then on the basis of that, and that you could also, ideally some of them would also be ecologists that would deliver independent assessments of, if you were to release these mice, is this likely to be effective enough for to be worth proceeding with the field trial? Now, one open question is, I don't know that you want to go all the trouble of having a formal vote at that point. So we could do something more. There are less formal polling methods. Anything from simple, whoever cares to show up probably cares a lot one way or another, and then they could be the ones that matter, to random anonymous polling boxes or what have you. So far, it's been pretty clear what the community wants just through meetings like this, insofar as those people who are interested enough and are to show up on a beautiful day like today.
Thank you, Chris Reed. One question that kept coming up in my mind was, you mentioned a gene drive and that this wasn't that, which made me want to know what that is.
So, all right, so a bit of confession. The reason why I am particularly interested in this project is because I am afraid of what happens if science continues on its route of being conducted behind closed doors. And that's because, bluntly, technology is becoming increasingly powerful, and I want communities to keep an eye on scientists like myself so that you can help keep us out of trouble. And that's particularly because 4 years ago, I had played a role in developing this CRISPR technology that's programmable molecular scalpel. We can direct it to cut DNA in any gene, any species, basically, and therefore edit it. And I realized, well, what happens if you program the editor in along with the change you want to make? And if you do that, then you make an organism that can edit DNA on its own. So when it mates with a wild-type organism, the offspring inherit one edited copy and one wild copy. But in this case, the edited copy has the editing machinery along with it. So it cuts the wild-type copy, then the cell copies over the edited version, so now it has two edited versions. Since it has two edited versions, it's guaranteed to pass it on into the next generation. And meaning that instead of half the offspring inheriting it, they all do. And then if they mate with a wild mouse in this case, it would happen again and again and again and again. It would double every time until you've affected all of the mice. Problem being that as soon as some mice take a ferry to the mainland, then it starts doing that there until eventually you've done this to all of the mice. So I am very concerned with ensuring that this technology is never developed behind closed doors by anyone, and that anything even remotely like this is only considered with the community keeping a very close eye on everything that is done from the outset. So this is a case where our laboratory is also working on ways of doing this in a controlled fashion. So that is setting a generational clock on it such that it can only do this copying itself step so many times before it runs out of other essential elements that it's losing every generation due to the normal inheritance rules. So we call this a daisy drive because it's set up like a daisy chain, and it loses one element from the end every time it copies itself. So then when it runs out, then it stops. And we're also working on ways of ensuring that, in effect, the genes themselves take a vote and would arrange such that whichever version is in the majority, either the edited version or the original version, would be favored. So that you could release these daisy quorum mice, say in the center of a town. It would spread the immunity out to near the borders. The daisy system would run out of elements. And then in the town, the edited immune version would be in the majority, so it would go to fixation. But whenever mice that were edited moved outside the town's boundaries, they would then be in the minority, and natural selection would eliminate them. So it's a way of letting you release many, many fewer organisms, but ensuring that the entire effect stays within any arbitrary political boundaries you want. That's, I think, one of the only ways that this kind of technology could ever be ethically justifiable. The downside, from many people's perspective, is that then you'd have to use DNA that is not from that particular organism. It actually is in the mice, but scientifically this is not a downside, right? You use the DNA from the bacteria in the mouse's gut. That is, in the sort of grisly metaphor, If you take a dead mouse and you put it in a blender and you extract all the DNA, everything you would need would be in there. That is, it's part of the mouse superorganism. But a lot of people feel very strongly about not using any foreign DNA. And again, it's up to the community. But the benefit is every mainland community then could be like Nantucket. You have the benefit of being an island. If you want to do this, it's not going to affect any other community unless there is something like a gene drive, which we don't think is a good idea.
Thank you very much.
Kevin, good presentation. Roberto Santamaria, the director of the health department here. You mentioned that some of the DNA will have 5 copies of each organism. Is the 5 copies the number that you're looking at as your target number? And then my second question is it going to be on the same autosome? More so for the fear of creating an unintended palindromic sequence that might end up with more DNA sequencing and recombinant DNA, things like that.
So the idea is to use CRISPR to insert as many copies as we can. And there's different ways of doing that we're going to have to explore. One way is to target several independent, safe loci in the genome, ideally on different chromosomes because we want them all to assort independently. Okay. So we want to ensure that on average, if, say, we can make a mouse that has 16 copies, then we want to ensure that they all assort independently, so on average offspring get 8, their offspring get 4, their offspring get 2, their offspring get 1, and so on, which means then we have to release many fewer mice. We don't really anticipate problems with recombination and rearrangement, just because if that were to happen, first of all, it wouldn't change the positions. You would have one at each position already.
Perfect.
And most rearrangements like that are pretty bad for the organism because you get a chromosomal translocation that then leads to problems interbreeding.
Okay.
So we don't think that's going to be a problem.
Perfect. Thank you.
Hi, I'm Joni Steele. I'm a summer resident, and I have gotten Lyme here on Nantucket before. My question is about unexpected or unintended consequences, and I was just wondering if you remove the main food source for the ticks, will they be more assertive or aggressive in finding other food sources? They seem like they're pretty good crawlers to me. They seem very quick, very fast. I've seen them, and just wondering maybe if they actually start attaching themselves to humans more because they're looking for a food source, or our dogs, or...
Great question. Sam, do you want to take that?
That's an excellent question, and in fact, it may be the key as to why we see lots of cases one year and few cases the next, is how many mice do you have around in June? Was it a bad winter and you have very, very few mice, which means that the nymphal deer ticks have nothing to feed on that they normally feed upon, and they go looking for humans, and humans are more likely to see them. But the possibility of making the mouse less likely to feed larval and nymphal deer ticks by heritably immunizing against one of the tick salivary proteins will, one, make the mouse reject the tick, but some of that rejection is by grooming. So they groom and they eat the tick off of themselves. But the other thing is that is already happening. These mice are communally nesting. They nest in nest boxes, for example, 6 or 7 of them together, and they groom each other. And so this is already happening out there, and the number of animals that are out there have not replaced mice when they're absent. We still continue to see ticks year after year after year, even if we have few mice. And there's been no, other than the June suggestion, the June correlation between mouse density and human cases, it doesn't imply anything for future generations of ticks and what they'll feed on. They seem to just go right back to mice when mice come back. If everything goes as planned, and all of this is going to be looked at by mathematical modeling, computer modeling as well, to see what we might be able to predict from, if you introduce this many mice and this many will be immunized against tick bites, what will happen to the tick population if we assume that there are these numbers of other kinds of hosts out there, what would be the effect of this. So all of this has to be planned out in detail, and that's one of the reasons Nantucket has been the target for these kinds of studies, is because we have a very rich database going back to the 1970s with the original Harvard investigations on Nantucket fever, which I am an intellectual offspring of. So I've continued the studies that came out of the Harvard lab where I did my doctoral degree, that they started in the 1970s. So we have a long historical amount of data that can be used to inform mathematical models. But until we do a field release, there's no way of really giving you the answer that you would like. But it's an excellent question.
And definitely one of those things where if you're concerned about that, then that's the sort of thing that we would need to study in the field trial and monitor post-release of the mice. Then are there more questing nymphs that have, say, already bitten a mouse and then fallen off and then are in the environment? They should show up on the other animals, or in this case on a tick drag flag.
On a tick drag, absolutely.
And so they should be detectable in that form. Now, it is important to note that if they haven't bitten an infected mouse as larvae, then the nymphs are not going to infect you with disease. Unless, of course, they've bitten something like, say, an infected chipmunk or the like. But
no chipmunks here.
That's right, no chipmunks in Nantucket. There are the shrews, though, so it could be a shrew. But there may well be more. We don't know. If there are, they would at least be less likely to be infected than the current ones. So,
thank you.
Your early thoughts about fundraising? I know it's early, but what are you thinking about that? Will there be perhaps a commercial application to this that could be a source of financial, finding companies, I don't know, who might support this financially?
So right now we are looking pretty much entirely at philanthropic fundraising for everything from the field trial onwards. And there seems to be a general level of interest in the project, primarily due to a lot of people who are concerned about tick-borne disease on Nantucket and the Vineyard, and also in other places, but also just people who are interested in open science and trying to change the culture of science in this way. So we think that philanthropic donations will suffice. This is frankly not the sort of thing that could be commercialized, just because it's probably not the sort of thing that most island communities, and again, it is only island communities without something like the Daisy Quorum Drive that I described. It's just not something you can do anything other than an island. And you have to release so many mice that it's probably not commercially viable. And again, I think keeping this nonprofit is a good idea.
Sorry, one last question for me. Is when you're making the immune mice to specific ticks, are you casting that wide net? Because I know we're having some invasive species like the Lone Star tick, the deer tick, the dog tick. Is it going to be effective against all of these other types of ticks as well?
Not likely. Although the molecule subolecin, the constituent protein in tick saliva, has what we call homologs in the DNA of these other kinds of ticks, such as Lone Star ticks or dog ticks. But sadly, they feed on different hosts. The Lone Star tick, all three stages rely upon deer, and to a certain extent on birds like grouse and turkey and so forth, and quail. And so you would have to do the same heritable immunization of the hosts of the Lone Star tick. The dog tick is virtually extinct on Nantucket. It's harder and harder to find out here, largely because everybody's now using preventatives on their dogs. And you have no raccoons and skunks, which are the other main reproductive hosts for dog ticks.
Perfect. Thank you.
Mary Longacre, sometimes you're saying Nantucket, sometimes you're saying Nantucket and Martha's Vineyard. What is the relationship for the two islands? Is it one or the other? Is it both at the same time? Is it one and then the other? I'm not quite sure where you're headed.
So right now we are, in effect, guided by two steering committees, one for Nantucket and one for the Vineyard, and the decisions are totally independent. It's also worth noting that it's a matter of debate as to whether or not there really are different subspecies on the two islands. but that means that the populations of mice that would need to be bred up for release on the different islands would in fact be separate. Even if they're not formally different subspecies, they are genetically somewhat different. And so they would need separate populations to be bred up and released. So it is separate decisions. It's running in parallel. Either both islands could decide to move forwards, neither, or one and not the other.
Maybe we're being devious and playing the two islands off themselves, like with the football rivalry.
So your project could branch at any point and take a slightly different path depending on what that island wanted?
That's right. That is, what the vineyard decides would not affect Nantucket because we would need to breed up different mice for you anyway.
And is there a funding implication to that?
Right now, we're not thinking of it that way, but I suppose you could say
And
a
you
funder could commit to only funding a project for one island and not the other, I suppose.
Well, I'm thinking, are you anticipating or building a budget based on attacking both islands? And if both of them did not have the same decision, would one be torpedoed because there wasn't enough money? Or is there any consequence to the two islands deciding differently?
I don't think so. Again, we have not attempted to fundraise at all from the philanthropic sense. So I don't know. I mean, the market could crash tomorrow and then, well, yeah, so much for that. But we don't think that will be a problem. And we're working out how to do that. I mean, it probably would be that any philanthropic gifts would be, of course, returned as soon as an island says no. Now, whether or not they might be earmarked for one island or the other or for both would depend very much on the donor.
So right now, would it be fair to say that you're being funded as a scientific inquiry and not as a public benefit where you're drawing money from public sources? You're just more in the general nature of things.
Well, so we are actually being funded by your taxpayer dollars theoretically soon, just on the because we've been recommended for this federal award.
Right, but that's coming
that's
equally from Nantucket or Martha's Vineyard. It's not,
well, and all the other taxpayers in the United States, yes.
Yeah. Okay, so we're still at a sort of generalized point here, and will remain there for a couple of years, probably, is what it sounds like. Okay.
And from our perspective, in terms of initially engineering the mice in the laboratory, it is not really particularly different to introduce one set of antibodies or both. We can do that separately. We can do both of those.
And then are there other potential locations where it would make sense, would there be a benefit to release mice in this manner?
Any island that has a lot of tick-borne disease.
But specifically islands.
Yeah, this approach is, on the mainland, if you release, say, 100,000 mice in a town, it would last a few years, and then it would just be diluted out because there's probably more than 1 billion white-footed mice on the mainland. So if you want to do something like that, then you need that quorum approach that then would keep the engineered genes present in the town and dominant, and therefore the local population immune. Without something like that, it's just not something that could work on the mainland.
I actually don't agree with that.
Oh, really?
I believe that we need to look at that with some mathematical modeling. But on the mainland, what we have is sort of mini islands that you have these islands of risk around communities. And so it's almost like the argument people have given me about using tick tubes. If I put tick tubes down around my home, but my neighbor doesn't, am I not just wasting my time because the ticks will come in from the other yards? And I'm going, no, you're protecting yourself in the immediate vicinity of your family. What other people do is not germane. It's you protecting your yard. And so I think you can actually protect blocks, neighborhoods, communities, by this kind of heritable immunization, even on the mainland, because the mice, if you turn them loose around someone's yard, it's not as if they're going to go a mile away and reestablish themselves. They're going to hang out around that area. And their generation after generation, they're going to hang around in that area. So I actually don't see this as being not applicable to mainlands.
Other than the sheer numbers argument, perhaps.
Maybe.
Although it is true that we could do just the quorum without the daisy drive part.
Yeah.
And then we could release 100,000 mice in a town, and then that would keep the immunity there. and robust against invasion from the wild mice on all sides, regardless of how the gene flow works. But again, that's 100,000 plus mice for every town that wants to do this in the entire mainland. It just doesn't really scale. But in theory, maybe.
And you mentioned that house mice are very different animals than white-footed mice. Do house mice host ticks, and do we have house mice on Nantucket?
I have looked pretty hard. The only house mice I've ever seen are downtown, around kitchens and things. But if you look in your homes, it's all white-footed mice.
Yes, okay.
So there are very, very few house mice here. The only place that had house mice as the host for ticks and Lyme disease was Cuttyhunk Island, where it once had the distinction of having no white-footed mice, but only house mice, even out in the bushes where they're not supposed to be. But white-footed mice have recently invaded Cuttyhunk from nearby Nashawina, and they now have more white-footed mice than house mice, and I've had a hard time trapping them on Cuttyhunk anymore. So it is true they could themselves serve to keep the cycle going, but in general they're very urban, they're very domesticated species.
And then I assume that your techniques would not work at all on the house mice because you're not building them for that species.
Well, so we are working on, so house mice are an invasive species here, and there are, on many islands, they are a destructive invasive species. Not quite so bad as in rats, but for example, they're driving some endangered albatrosses extinct in some tropical islands. So one of the things that we're hoping to do with Daisy Drive is we can design it to suppress a population by interfering with fertility. And because of the daisy effect, it would be a local population only. It would not be able to spread off the island. And so we're looking at this as a possible conservation tool for saving endangered species by removing mice or rats from an island. So we're looking into building these systems in both house mice and in rats. So, and we would love to talk with interested Nantucket citizens because you do have rats, yes?
Yes. Mm-hmm.
So if you want to rid the island of rats at some point in the future, this is something that we should discuss. This is not, rats are not threatening any native species with true extinction, but some of the threatened terns, for example, I believe, are damaged by the rats, and, well, rats are not among, they're probably the third worst invasive species in the world. We are number one, the domestic cat is actually number two, and the rat is number three, at least when it comes to causing extinctions worldwide. So we're very interested in this, and most of the tropical islands where this would do the most conservation good don't necessarily have a lot of people living there. So this is another technology where we would love to have a community keep an eye on it, with an eye towards potential future use here, maybe someday. Maybe never, but if nothing else, we would love to have you keep an eye on the project and tell us what we're doing wrong and what we need to look out for that one as well.
All right, thank you.
I just have one quick question. Oh, go right ahead. Yeah. Other than a forum such as this, and you started out your presentation with the buy-in or the opt-out of the community, what are like the next steps? I mean, this is the first time I've heard of this. I've read about it in the paper, and I'm here. But what are there going to be more forums like this, or is it going to be going on a more town level? And what are your next steps?
So that is a great question, and part of that is, if you have any suggestions for how we should be doing it differently, then please let us know. Because again, we're making this up as we go along. There aren't a lot of examples of projects that really put the community in the driver's seat, and that's sort of what we want to figure out. What is the best way to do this? Mm-hmm. So there will definitely be future meetings, and at least two, probably 3 or 4 a year. There will also be steering committee meetings. So the steering committee members, majority of them live here, at least part-time, most of them full-time, and you can talk to them, and we will probably have at least more regular meetings, at least by Skype. So raising concerns and things that need to be looked at. Our site, Responsive Science, has a project page for this project, and we're going to be adding information. So laboratory advances, Joanna's recent advances, which are in fact very recent, which is why she's been staying up late to generate them, will all be put up there, as will additional advances. And the site is designed to encourage commenting, so we hope that there will be a robust forum in that form. Obviously, a lot of people already use Facebook, and there's a Nantucket Facebook page if you want to. We'll be monitoring that. So if you say, hey, we really hope the Mice Against Ticks, or whatever you want to call it, project needs to look at this, then feel free to add it there, and there can be a discussion there.
Do you foresee a vote? I mean, or you have to, right? I mean, if you want the community buy-in.
So eventually, before mice can be released on Nantucket, there would need to be a warrant at the town meeting and then a vote. And so that will be the ultimate decision, will be a final vote.
There will be no genetically altered mice on the island unless it goes to town meeting.
Right, okay.
Mm-hmm.
So you're building
Absolutely.
up to that. Thank you.
Anybody else? I have a question. Do you view the fact that the vineyard is somewhat more politically diverse with 6 towns, do you see that as being a potential problem trying to get a consensus around going ahead with this, as opposed to a sort of single person government here, single person?
Nantucket does seem remarkably efficient in terms of making decisions and making things happen. It is true, whereas I gather that the vineyard still cannot decide what to put on their new fields as to whether it should, what kind of turf or natural grass or whatever that it should be, which is maybe suggestive. On the other hand, they have a severe problem with tick-borne disease as well, and all the towns have appointed members to the board. All the town boards of health on the vineyard have appointed members to the steering committee. But yeah, getting 6 towns to all vote yes is probably less likely than 1 town voting yes. That's just mathematical. Yeah. All right. Finally, I just want to say thank you all so much for coming. It really is an honor and a privilege, especially because have a lovely evening.